So for, as Luna mentioned, and thanks again for inviting me this year as well, I'm gonna talk about some application of the fluorescent fluctuation techniques that you learn during this workshop so far, applying this on potassium channel subunit dynamics and stoichiometry. So an overview of the work that we've done with Lorenzo that you met before, and LFD and Abbott Lab from UCI as well. We use CHO cells, so Chinese hamster ovary cells. We transfected or co-transfected them with fluorescently tagged potassium channel subunits. We used total internal reflection microscopes to get our data set, since this is like a membrane channel, so we were interested in acquiring data only from the membrane. And then we applied on the same data set 3 of the different techniques I can show you. I'm showing here: MSD, number mRNAs, and 2D PCF. So a little bit of background on the system that we're using and that Abbott Lab is an expert of. So we're using voltage-gated potassium channels that are formed by alpha subunits that forms a pore, and each of these subunits is regulated by the interaction with other beta subunits from the KCNE family. Some of these channels, like the one that we're interested in, Q1, are active constitutively at the resting membrane potential and also have a role in non-excitable cells, such as epithelial cells. In particular, this complex that we're interested about, Q1E2, is an epithelial complex that is essential for the normal gastric, thyroid, and chloroid plexus function. And we look into this because there are still some not completely addressed question about the molecular composition, the stoichiometry of this complex, and the different subunit dynamics. So the first thing that we wanted to check, and Abbott Lab focused on this part, that the fluorescent tag that we put into the system didn't affect the channel functionality. So to do that, they measured the current generated by the tagged version of Q1, and they showed that they were similar to the currents that they were observing for the untagged KC and Q1. And then they showed that the complex, the tagged complex with both Q1 and E2 fluorescently labeled, were constitutively active, and they were expressing smaller currents compared to the Q1 tagged. And that is similar to what they observed for the untagged complex. So once we were sure that the fluorescent tagging was not affecting the functionality of the channel of interest, we took these samples and we put it into our system. As I mentioned before, is a total internal refraction fluorescence microscope. So since we were having a double tag with MTRI and GFP in a system, we have like a double laser excitation. So one laser to excite the GFP and one white laser with excitation filter to excite the MTRI. And then, as I mentioned before, this particular way of illumination is highlighting only what's happening at the membrane of the cells. And then for the acquisition, we were using a CMOS camera. Just a little bit of background on the different techniques that we're using. So as I mentioned at the beginning, and Lionel mentioned as well, we were acquiring one data set, and then we were able to analyze the same data set with these 3 different techniques. So the first one is MSD, that you probably already heard about. So this analysis uses spatial temporal fluorescent fluctuation caused by the protein diffusion, and gives an idea of the protein mobility in the nanoenvironment. So this function that you can see here, from this function, you can get different kind of parameters. In particular, you can get the length of confinement. So it means the size of the domains in which a protein is diffusing. you can calculate the diffusion coefficient inside these nanodomains or across the nanodomains. Here you can see the macro, and also the diffusion modality. Here is a representation of the three principal kind of modality of diffusion. So a free diffusion, a confined diffusion inside some domains, or a transiently confined diffusion. So once we analyze our data with this technique, we saw that both KCN-Q1 and KCN-E2 were showing a transiently confined mode of diffusion on the membrane. And this was happening whether they were independently expressed or co-expressed. Then we went further and we measured all the parameters that I mentioned before. So in particular, we observed that KCN-E2, when it was co-expressed with KCN-Q1, was showing two different populations. So we separated these two populations, and we observed that E2, the slow diffusion population, was having a similar behavior to E2 when it was expressed alone in the microenvironment. But a longer spatiotemporal range, both these populations were more similar to E2 when it was expressed alone, while this population here was more similar to the behavior of Q1, the short range. So our hypothesis was this was representing the unbound population, while the fast population was representing the one that was bound to Q1. Then we want also to check that in the range of expression that we consider, these parameters were not dependent on the expression levels. And we saw that at least in our range of acquisition, there were no dependency on the expression level since it was a transient transvection. The second technique that we applied to our data set is the 2D PCF that Francesco just talked about. So briefly, we can use this technique to measure the directionality of the protein diffusion. And in this case, if you apply this to different angular direction, you can create a map of diffusion by cross-correlating different pixels. In particular, we can use eccentricity to measure if the protein is encountering obstacles or not. So eccentricity higher than 0 means that the diffusion is no longer isotropic. And also, this analysis can be carried out at different pair correlation distances. In our system, we use 5 different distances. And here you can see the curves that we obtained for the different per correlation distances, the values of eccentricity as a function of the per correlation distance. And here you can see the intensity image of the cells in the different conditions and the eccentricity maps for an example of 3 of the per correlation distance considered. From the data, we can see that KCN-Q1 show really low eccentricity that is comparable with our inert control. So our inert control that is here is just a membrane-bound form of EGFP. Then we observed that a longer distance, the fast and slow E2 bound population, E2 Q1 population, and were similar in behavior as E2 with higher eccentricity compared to the Q1, while a short range, the fast population of E2 seems to localize in the membrane in regions with more pronounced motion directionality. The final technique that we applied in the same data set is number and brightness that you heard Enrico talk about this morning or afternoon. So in particular, from this technique, calculating the average and variance of intensity distribution for each pixel, we can determine the concentration, meaning the number, and the aggregation state meaning the oligomerization of the proteins. For this technique, we need to use as a reference the brightness of the monomer. In our case, as I mentioned before, we were using our inert control, that is the membrane-bound form of EGFP. So in this case, using this technique, we get some more information about this system. In particular, we saw that E2 exists as a dimer. When is co-expressed with Q1, we can have for this low population a mixture of dimers and tetramers. And when we have the fast population, we have tetramers. So our interpretation of this is that the dimeric state of E2 represents the unbound form, while the tetrameric represents the one that is forming a complex with the Q1 subunit. You can see here that Q1, whether is expressed or co-expressed with E2, shows a tetrameric complex on average. That is what is reported in literature, of course, with other techniques. So that was the confirming that our analysis was correct. One thing that I like to mention, since all of these techniques are from all of this analysis, you can build some maps. You can, and it's done on the same data set, this kind of analysis. You can also have more insights about what's happening in particular region of interest in the cell. So here, there's an example on the same cell. We have the 2D PCF map, and we have the brightness map. So for example, in this region here that I highlighted, we can see that this region have like a really high eccentricity, meaning like a pronounced directionality of motion, and this also corresponds to having like a higher brightness. So from this, you can have the opportunity to further explore particular region of interest in your cells, and you can also have maps for the MSD as well. So what we did to try to gather all this information together, we tried to plot as spider plots to represent the different parameters that we can obtain by this multiplex analysis to show the differences between the different subunits and conditions that we have. And also we were trying to do like a schematic representation. So here you can see for example, the confinement are the size are related to the size of these squares is related to the length of confinement that we can get from the MSD. See, from MSD, we can get the micro and macro diffusion that are represented by the dark blue and light blue circles. We try to represent the directionality of the motion by these two ellipses, what we can get from the 2D PCF analysis, and also here is represented the average oligomerization state that we can get from the NMB analysis. So in conclusion, we found that this complex, this is an octamer formed by 4 subunits for Q1 and 4 subunits from E2. And we show that every subunit has a different particular dynamic signature. And this can improve the understanding of the physiology of these channels. And also we demonstrated that using together turf imaging in combination with this fluorescence fluctuation spectroscopy techniques is a really powerful tool, and it allows to determine with a single data set acquisition, polymerization state, diffusion modality, and the nano-environmental organization. So this can be applicable to not only this field, but for, of course, like every membrane protein, like for example, receptors, other kind of channels, and so on. And it can also be used to monitor, for example, effect of drug treatments and so on. So with this,